Welcome back, biology, and today we're going to start our discussion on chapter 12, which is inheritance patterns and human genetics. So what we're going to be talking about in this chapter is human genetics are a little bit more tricky, as we talked already, than pea plants and other simple organisms that have kind of simpler Mendelian genetics. So we're going to look at things like this as pedigree here, and we're going to look at how traits are carried from grandparents all the way down to grandchildren. And we're going to look at different modes of human inheritance and how these things are transferred and why humans are a little bit different. All right, so the first thing we need to talk about is chromosomes and an early work of chromosomes. In the early 1900s, Thomas Hunt Morgan, he was examining and experimenting these guys, the fruit fly, Drosophila melanogaster. And these guys are a nice model organism. They work really well for studying genetics because they only have 4 pairs of chromosomes. And he noticed that when he looked at these chromosomes under the microscope, 3 of the 4 pairs of chromosomes were all the same size in all individuals. His research showed, however, that the 4th pair was different. In females, they were identical, but males had 1 large chromosome and 1 small chromosome. He called these chromosomes the X and Y chromosomes. We now refer to them as sex chromosomes. So there's some difference between the sex chromosomes and the other chromosomes, which we call the autosomes. The sex chromosomes, these determine an individual's sex. An autosome is what they aren't determined in this determination of sex. They are the ones that carry genes that aren't sex related. So in human males, they have different sex chromosomes. They have XY, and in females have identical ones in humans, which are XX. Other animals aren't always the same. Animals such as chicken and moths, the males have the same chromosome, which is YY, and the females have a different chromosome. They have an XY. So it depends on whichever species and group of animals you're looking at. And for sex determination, like other homologous chromosomes that are splitting off during meiosis, the sex chromosomes pair during meiosis 1. They then split up and move to different cells. Because of this, the sperm have an equal chance to have a X or a Y chromosome that they're giving. It's because men have the XY genotype, so they could either give the X or they could either give the Y. For our females, who are always XX, it doesn't matter what they give, it's going to be an X. So in mammals, it is the male organism and the sperm of the organism that determines what sex it will be. And how this happens is through a region called the SRY region, which is a gene on the Y chromosome that helps for sex determination. So like we said before, when a male gives the X chromosome, the offspring will be female. When it's a Y chromosome, the offspring will be male. This Y chromosome holds this region called the SRY gene, which is the sex determining region Y, SRY. And this codes for a protein that makes the gonads develop into testes. All embryos start out female, and then the SRY region kicks on with testosterone, and this makes an organism male or female. So the effects of a gene's location affect how it's displayed in an organism. When working with fruit flies, Morgan replicated Mendel's methods. He carefully crossbred flies with certain traits. He did the pure finding kind of method where he would take a pure breeding, true breeding red fly and mix it with a true breeding white eye fly. And the fly that has white eyes is the little e, and the one that has red eyes is the big E. Red eyes is dominant. So in the first generation, as we would expect with the dominant trait, all of them were red eyes. And in the second generation, the F2, there was a 3-to-1 ratio where only males had white eyes, this little e. And try as hard as he could, Thomas Hunt Morgan could not breed female flies with white eyes. What this told him is that some genes are sex-linked genes. So he thought that the gene for eye color must be located on the X chromosome here. And so a sex-linked trait is any trait that is coded for by an allele on a sex chromosome. And because there was no counterpart here on the Y, there was nowhere for that gene to have another copy. So there was not 2 alleles. Remember that alleles are, like when we're looking about big A, little a, each one of these is an allele given from another parent. So something to note is usually the size of the sex chromosomes is different. In mammals, the X chromosome is much larger than the Y chromosome. If they are on the X chromosome, if genes are on the X chromosome, they are said to be X-linked genes. Since we only have one X chromosome, if there's a gene here or here, there's no counterpart on the Y, so that gene is the one that is expressed. So an example is colorblindness is carried on the X chromosome. So if you are a male who has one copy of colorblindness, you are colorblind. If you're a female who has one copy of colorblindness, you still see in color because you don't have two copies. You have another part over here to kind of back that up. All right, linked genes. If genes are really close together, they can be inherited together. And so linked genes are pairs of genes that tend to be inherited together. So they're always on the same chromosome. These guys are on the same chromosome. And they're usually close together. So like these two, they're not linked. They're farther apart. And these two, also not linked. They're really far apart. These two are on different chromosomes. Over here, this one is really far apart on the same chromosome. All right, crossing over. During crossover, we have pieces of chromosome break off and attach differently to their homologous chromosome. This creates exchanges between chromosomes that increase the genetic variability. So if the blue one's from dad and the red one's from mom, now you have a brand new chromosome that has a piece of mom's DNA on a dad's chromosome that has never before been seen in the human population. So this is why humans have such great genetic variabilities. We're constantly crossing over and mixing and melding all of these different chunks of DNA. The important thing with crossing over is it always happens between homologous chromosomes. So those that are, both of these would be chromosome 1, or they could be chromosome 2, but you couldn't have one that was chromosome 3 and one that was chromosome 4. Chromosome mapping. The farther apart two genes are, the more likely a crossover will happen between them. So the less likely they are to be inherited together. A chromosome map here shows the linear order. On this one you have body color, and then eye color, and then wing size. And when we have these, we'll break them up into map units, where one map unit is a 1% chance that the genes will cross over. As we move further and further away, this percent goes up. So the difference between here and here is only 25%, where the difference between this guy and all the way up to the top there is 45%. So it's more likely that a crossover will split this up, and this chromosome will be inherited separately, and this part of it, and this one will be inherited on a different one. All right, mutations. So a lot of the discussion going forward in this class is going to be about mutations, and this is the change in the nucleotide or DNA sequence of a gene. In germ cell mutations, this is what's happening in an organism's gametes, and it can't be passed, it has to be passed on to an offspring. It doesn't happen in that organism. For somatic cells, they take place in the body cells of an organism, and that organism lives with those mutations its whole life. Mutations that cause death, often before birth, are lethal mutations. Lethal meaning, of course, deadly. Some mutations, however, are beneficial to an organism. They stay in the population. In humans, things like having blue eyes is a mutation. It's not detrimental. It doesn't kill you. Some people think that blue eyes are pretty, so it is somewhat beneficial. So this mutation has stuck around inside of the human genome. There are different types of mutations. The first are chromosome mutations, and these involve change in the structure of actual chromosomes. And in the first one here, we have deletions, which is the loss of a piece of chromosome. So in this mutation here, this original chromosome loses this blue piece to become mutated. And that's a whole lot of genetic information that was just lost. The second type is inversion, where the chromosome section breaks off, flips around backwards, and reattaches. So this one was reading this way, and now all of a sudden it's reading backwards. So this will also change how this chromosome is read, because now when this amino acids come through and they make the proteins will be different. Instead of being 1, 3, 5, 2, now they're 2, 5, 3, 1. And that could change the protein structure and therefore change its function. Translocation is a lot like crossing over, where a piece of chromosome breaks off and then reattaches, but it's to a different chromosome. one that it's not supposed to be attached to. So if these are chromosomes 1 and 1, and these are chromosomes 2 and 2, this chunk that's breaking off down here and switching over is not supposed to be doing this. So this is a translocation. The fourth and final kind of chromosome mutation is nondisjunction, where chromosomes fail to separate from their homologous pairs. What happens is one gamete receives an extra copy, this one has 2, and this one receives no copies, it has 0. An example of a disorder caused by this is Down syndrome, which is trisomy 21 or 321, so it has 3 chromosomes here, because what will happen is these 2 chromosomes here will mesh with one from mom, and now we have 3 chromosomes inside of this newly formed cell. All right, gene mutations. These are ones where we have a point mutation, where it's the substitution, addition, or removal of a single nucleotide. And in a substitution, one nucleotide replaces another. So an A could be turned to a C, or a T could be turned to a G. In deletion, one or more nucleotides are lost or removed from the sequence. And in an insertion, one or more nucleotides are added to a gene. So looking at this normal DNA here makes this mRNA, which then makes these amino acids. In this first example here, we're substituting this C for a T. When this happens, now instead of making phenylalanine, tyrosine, instead of making glutamine here, we now make lysine. So we've changed this amino acid structure, which probably changes the protein structure, which will change the protein's function. In an insertion, we are adding another C in here. So now instead of CTT, it goes CCT, TCT. And this other C is like displaced over here somewhere. Probably like right there. Oh, excuse me. Probably like right. Probably like right there. So this not only changes one, but changes 2. Changes this one here to glycine and this one to arginine, which is a lot different than this original one. And then the last one here, this A originally is deleted. And so everything is shifted over, and now instead of having phenylalanine, tyrosine, glutamine, we now have phenylalanine, threonine, lysine, arginine. So this is a really big shift compared to the original structure. Whenever we get these shifts in the reading frame, whenever it changes so that their codons are read incorrectly, we call this a frameshift mutation. So when this happens, like this one is a deletion, this causes the wrong protein to be coded for. Instead of methionine, glutamine, alanine, glycine, it's now methionine, lysine, histidine, and then this one's not even there. So this one's smaller, and it's the wrong amino acids. So frameshift mutations are the ones that are usually pretty bad, usually your lethal mutations. All right, moving on to 12.2 is all about human genetics, starting with pedigrees. And a pedigree is this kind of diagram here that shows how a trait is inherited over several generations. On it, the squares represent males, and these circles represent females. Additionally, a circle or square that's filled in, like these ones here, means the individual has a condition or has a trait that we're looking at, whether it's blue eyes or color blindness or Huntington's disease. If there's a horizontal line between 2 individuals, female and male, that means that this is a mating pair, and then the vertical lines are their offspring. We go from the oldest, the first one, to the youngest, which is the last one. Additionally, we have these Roman numerals here that label which generation we are. If these guys were to have kids, this would turn into generation 3, and this would continue on and on and on, as many generations as you can go back. Now, humans have a little bit different patterns of inheritance. And so this kind of study is the expression of genes over generations. So we're looking at grandparents to parents to their grandchildren, and so on and so forth. Things that are autosomal, or body cells, happen equally in male and females, and sex-linked usually are only seen in males, and sex links are usually recessive genes. So we usually see recessive kind of patterns as those are being passed along. The first pattern we're going to look at is autosomal dominant. An autosomal, remember, is non-sex related chromosome, and dominant means that there's only one copy that needs to be expressed. The cool thing about autosomal dominant is every individual with this trait has at least a parent with this trait. So like this guy here, number 3, he has a parent, his mom here, who gave him the allele for this. His dad doesn't have it, but his mom did. So he gives this, and then he gives the allele to both his daughter and his son. See, both males and females are getting it from dad here. Mom doesn't have it. And then on the other side here, we have the daughter, who is then giving it to the grandson through this trait. The next strategy is autosomal recessive. This is one where a child can have 0 parents, 1 parent, or 2 parents who show this trait. If a parent also shows this trait, they are homozygous recessive, because this is a recessive gene. So like this guy here, neither of these parents have the disease, yet their son is able to contract it. If they show the dominant trait, they can be homozygous dominant or heterozygous recessive. So what this means is that these guys here are both, let's say it's Fs. They're both big F, little f, because they don't have the disease, but this guy has to get an F from the 2 little f's, and it has to get it from somewhere. So he got it from here. Same thing with this guy. He has to have little f, and this guy has to have little f, little f. Now, whether or not they pass it on to these offspring is kind of tricky to say. We know that eventually this shows up again, so I would assume this one has a little f from the parents, but that's just looking at it, having seen a lot of these. So individuals that have a copy of a gene but do not have the disease, we call carriers. And carriers usually get like this cool little like red circle inside of them where they're not all the way filled in. Like these two are obviously carriers because they can give offspring that, like this guy, this one is a carrier as well, and this one's a carrier. They can give offspring that has the disease. These both are carriers. This one's here's a little tricky because we don't know where it got the disease, the gene. It could have gotten it from the father here, or it could have gotten it from the mother. So carriers have the ability to pass the gene to offspring, and a lot of the time women will be carriers for sex-linked traits. Looking at how we would use one of these pedigrees to actually solve a problem, if we wanted to figure out what the genotype of this mother and this daughter was, we could figure it out. One thing that we know is that this daughter over here has to be little o, which means that mom has to be big A, little o. She had to have given this little o here. On the flip side, we see that down here, this offspring has a B, and this father is going to be little o because he's the recessive. So this has to be BO. So we know that this B had to have come from here. And we don't know what the second one is, so on this one, so we're going to put a question mark because it could be either an O or it could be an A. Now, looking at father here, we know that father has to be BO because he has to give the B to this child, and he gives the O to this child. So looking at this, this is the father and the mother's genotypes here. They would have a 25% chance of giving an AB individual, 25% chance of having an AO offspring, 25% chance of having a BO offspring, and 25% chance of having this O offspring. So these tables can give us a lot of information really quickly. So that's really, really nice. Looking at genetic traits, we can talk about different things. The first, we can talk about disorders. We can talk about how they're spread. The first thing we're going to talk about is polygenetic inheritance, where this is a genetic that's influenced by several different genes. Skin color, for example, is influenced by the effects of 3 to 6 genes. One allele determines whether you have more melanin produced. The other is less melanin. The other traits that we can have that are polygenetic are eye color, height, and hair color. This here is for growth. You'll see there's 64 possible genotypes here. And there's a big range between really, really tall people and really, really short people based on all of these genetics over here. So your parents give you a lot of information on these ones, and then that all kind of adds up into who you are as the individual. All right, complex characters. These are influenced both strongly by the environment and their genes. So in this example here, both of these tomatoes are off the same plant. They have the same ability to become this big, ripe, red, beautiful tomato here. But for some reason, this one didn't get enough sunlight or it was malnourished. It did not become a big tomato. the same genes, but the environment that it was in did not allow it to grow to its full potential. So, complex characters influenced both by environment and their genetics. So, in humans, skin color is a complex character. Exposure to sunlight causes your skin to become darker. No matter what your skin color genotype is, your body naturally produces more melanin. Some other examples are human height. Breast cancer is a complex character. Diabetes, stroke, schizophrenia are all examples of things that can be influenced by both the environment and the genetics of the organism. All right, multiple alleles. Genes that have more than 3 alleles are said to have multiple alleles. And they can have many different strategies. We've seen codominant already, where both alleles are expressed in the phenotype. So both of them get the capital letter. Remember that in blood type, it's the I. So IA, IB, and then little I is the recessive that turns into O. So if they are both IA and IB, then the individual shows characteristics of both. For our O-type blood here, it would be little I, both of those recessive traits. In incomplete dominance, we have 3 phenotypes for 2 alleles. If they have the 2 dominant alleles, they get the dominant trait. If they get 2 recessive alleles, they get the recessive trait. And if it's a heterozygous allele, the dominant and a recessive mix, it turns into some kind of intermediate trait for whatever that thing is. Our next inheritance pattern is on sex-influenced trait, where both males and females can have different phenotypes, different appearances, even though they have the same genotype. So these things are usually autosomal, because if they have equal opportunity to get them, they have the same genotype. An example of this is male pattern baldness. To show male pattern baldness, testosterone is required. So females don't produce enough natural testosterone to show this male pattern baldness, so that's why they don't get the receding hairline that we see in a lot of men. But surprisingly enough, usually this is X-linked. This is a weird one. So usually if you're a man, you need to look at your mother's father, and if he was starting to lose his hair, then there's a big chance that you will also lose your hair. This next kind of inheritance pattern is on single allele traits, where only one allele is needed to show a trait. Usually these are autosomal dominant things. Huntington's disease is a really good example of this. This is a condition that's characterized by forgetfulness and irritability. And every individual with this disease has a parent with this disease too. Huntington's usually shows up later in life, so people usually have kids and then in their 40s discover that they have Huntington's and that they probably pass that on to their offspring. And also, in addition, this is usually a trait where men get male pattern baldness, females usually get Huntington's disease. Detecting genetic disease. Like any other condition, medical condition, we want to detect these kind of diseases and be able to treat them. So we've designed and developed tests that allow us to test individuals for genetic conditions. This can be done by extracting blood, by testing for proteins that are made from the DNA, or by directly extracting DNA and running tests on the DNA. If we're worried about if we have parents with genetic disease, sometimes for infants, we will extract some amniotic fluid in the placenta, and we can examine the DNA of the infant from that, and then kind of see if these guys have genetic diseases or not. This is getting more and more common. It's getting cheaper and cheaper to do, so probably in your guys' lifetime, you'll start to see more and more couples starting to receive this kind of testing. And the last topic today is gene therapy. And in gene therapy, a healthy copy of a gene is implanted into the cells of a person with a defective gene. This is usually done by using a virus that injects materials into the target cells, kind of like those bacteriophages we talked about in an earlier chapter. And there are 2 types of gene therapy. There's somatic cell gene therapy, which affects only body cells, and there's germ cell gene therapy, which affects egg or sperm cells. Like before, somatic cell therapy would affect the individual that is getting the therapy, and germ cell gene therapy would affect the individual who is being born from that person, offspring of that person. This is the conclusion to Chapter 12. I hope you learned a whole bunch. If you have any questions, let me know, and I'll see you in the next one.